Good morning, students. Today we will learn about infertility and assisted reproductive technologies. Now, infertility refers to the stage when couples are unable to produce children. That means it is the failure to conceive even after 1 to 2 years of regular, unprotected sexual cohabitation, and such a phenomenon is termed as infertility. Now, this term is not synonymous with sterility, which means complete inability to produce offspring. So, infertility can be best defined as relative sterility. It is of two types. Primary infertility refers to infertility found in patients who have never conceived. That means when a woman is unable to ever bear a child. And secondary infertility, when a woman has been pregnant but failure to achieve live birth after having a live birth previously. Now, this can be due to number of reasons like physical, immunological, psychological, certain diseases and drugs. There are various causes of infertility in males and various causes in females. Now, in females, it can be due to ovulation disorder, it can be due to defect in fallopian tube, or it can be due to uterine or cervical defect. That means defective vaginal growth or the defects in cervix. Okay, so ovulation disorder includes no ovulation or oligo-ovulation. Okay, that means premature ovarian failure, inadequate growth and functioning of corpus luteum, which results in reduced progesterone secretion and which inhibits implantation. Sometimes the ovum is not liberated but it remains trapped inside the follicle. Fallopian tube may fail to pick up ovum and may have impaired motility, loss of cilia and blocked lumen. Sometimes there is defective uterine endometrium due to reduced or excessive secretory activities. Now, male infertility includes testicular causes, hypogonadism and other causes like drug addiction, like cytotoxic drugs and antidepressant drugs suppresses spermatogenesis. Many other causes includes cryptorchidism or failure of testes to descend into scrotum, absence or blockage of vas differentia, okay, hypothermia or higher scrotal temperature due to varicocele, that means varicose veins, low testosterone, low sperm count or abnormal sperm morphology. Now, any defect in sperm count, sperm structure, sperm motility of seminal fluid leads to infertility. Now, low sperm count is called oligospermia, while near absence of sperm is known as azoospermia, and defective sperm morphology is termed as teratozoospermia. Okay, so there are many causes. Now, some of these infertility disorder can be cured through corrective treatment. However, there are some disorder where such corrective treatment is not possible. So, in such cases, couples are assisted to have children through special technique commonly known as assisted reproductive technologies. These technologies involve surgically removing egg from women and combining with the sperm in the lab. This not only refers to IVF but also to several variations tailored to patient unique condition. Okay, and it is an experimental procedure, and these procedures are usually paired with more conventional therapies such as fertility drug to increase success rate. So, some of the prominent techniques are intrauterine insemination, in vitro fertilization, embryo transfer, intracytoplasmic sperm injection, zygote intrafallopian transfer, and gamete intrafallopian transfer. Now, in vitro fertilization means a technique in which the ovum is fertilized using sperm outside the body, and then the fertilized ovum is implanted in the female body, that is uterus, for further development. Okay, so IVF, it is an experimental procedure, okay, where doctor joins a woman's egg and a man's sperm in a glass dish in lab, okay, and then the fertilized ovum is implanted in the uterus of female for further development, okay. And this test tube baby refers to the conceiving of a baby which is produced in a culture dish and nursing in the uterus. Okay, that means a baby produced by conceiving in a culture dish and nursing in the uterus is called a test tube baby. It is not reared in test tube. So, and a scientific term for this procedure is in vitro fertilization, that means in glass. Okay, so the condition is similar as that in the body, which is followed by zygote or embryo transfer, and the same condition is then brought up or it is made in the lab, where fertilization of male gamete and female gamete outside the body is done. Now, test tube baby program. So this method involves in vitro fertilization, that means ova from wife or donor and the sperm from the husband or donor are collected and induced to form zygote in lab, and this process is called in vitro fertilization. Okay, so the zygote or the early embryo, that is up to 8 blastomere stage, is then transferred into the fallopian tube, which is popularly known as zygote intra-fallopian transfer, that is ZIFT, or embryo with more than 8 blastomere is transferred into the uterus, which is popularly called intrauterine transfer, to complete further development of zygote or embryo. Okay, and with proper medical care, the mother will give birth to a normal child on completion of gestation. Okay, now the first test tube baby was born in England on 5th July 1978, and it was a girl named Louise Joy Brown. And the first test tube baby in India was born on 6th August 1986 in Mumbai. Now, this is in vitro fertilization process where women's egg cells are combined with a man's sperm outside the uterus, and then the fertilized egg is then implanted in the women's uterus, and if it is successful, then it begins the pregnancy cycle. First, the woman is given hormone treatment to stimulate egg production. Then multiple eggs are taken from the women's ovary, and in the lab the eggs are mixed up with the man's sperm cell in a cultured dish to become fertilized. Now the fertilized egg or the embryos are placed in the incubator for about 48 hours, and then the embryos are implanted in the women's uterus or frozen for future implanting. Now this IVF is very important, and it has been a boon to infertile mother, or if it can be also treated with in men with low sperm count, and embryos can also be frozen and used for future. Now, zygote intra-fallopian transfer, that means the zygote or the early embryo with up to 8 blastomere is transferred into the fallopian tube. Okay, egg cells are removed from the women ovaries and then it is in vitro fertilized. The resulting zygote is then placed into the fallopian tube by the use of laparoscopy, and its success rate is 64.8% in all the cases. And because the fertilized egg is transferred directly into the tube, the procedure is also referred as tubal embryo transfer. Okay. Now, embryo transfer means it refers to a step in the process of assisted reproduction in which embryos are placed in the uterus of the female to establish the pregnancy. That means here also embryo with more than 8 blastomere is then transferred into the uterus. Okay, that is intrauterine transfer, and to complete the further development of the embryo. Okay, in this the embryo can either be fresh or frozen. If they are frozen, then they can be thawed right before they are transferred. Okay, now next is gamete intrafallopian transfer. Now, transfer of an ovum collected from a donor into the fallopian tube of another female who cannot produce one but can provide suitable environment for fertilization and further development. Okay, so it is useful when fimbriae fail to capture ovum and females having sperm antibodies in their cervical secretion. Okay, then this condition is adapted. Okay, so in this cases ovum from the donor female is surgically removed and is then introduced into the fallopian tube of such females. Okay, now next is intracytoplasmic sperm injection. So this is another special technique to form an embryo in the lab in which sperm is directly injected into the ovum placed in the nutrient broth, and then the zygote or the early embryo is later transferred by Zift process in women. Okay, so sperm is directly injected into the ovum in culture medium. Okay, so one of the partner's sperm is placed inside the egg with a microscopic needle rather than many sperm positioned close to the outside of the egg. Okay, as in IVF in a dish in a lab, and once the fertilization occurs, then the resulting embryo is placed into the uterus. Okay, now artificial insemination. If the infertility of male is either due to inability of the male partner, okay, to inseminate the female or very low sperm count, okay, then the semen of male partner or donor is collected, concentrated and introduced into vagina or uterus of the female, and this is called intrauterine insemination. Okay, so in this case the semen of the husband or donor male is first collected and then it is artificially introduced into the vagina or the uterus. Okay, so all these above techniques requires very precise handling by specialized professionals, okay, and expensive instrumentation. So apart from having many importance, these all have some of the drawbacks also. So these techniques are not possible with female with damaged uterine wall. Okay, and it requires high precision, specialized professionals and expensive instrument, and some it has been raised several ethical, emotional, religious and moral issue in the society also. Okay, so with these we came to an end of this chapter. Okay, so kindly refer to your NCERT and learn this chapter and whatever the topic has been covered. So that's all for today. Next we will be starting with the genetics. So keep learning, keep revising, and that's all for today. Thank you.